Just for all the speakers, I'll be sitting up here with you. We give you a 2-minute warning, and then you're done. I'm just going to come up and stand right next to you when you're done. And I'll grab the laptop if I need to. So we have 2 other sessions going on at the same time, so we're going to try really hard to keep everybody on the same schedule. So with that, here's Will Seamans from Kovalskill going to talk about panfish. Yeah.
All right, how's it going, everyone? My name is Will Seamans, and I'm going to be talking about my senior capstone project, looking at the diet, growth, and morphology in a wild population of pumpkinseed green sunfish hybrids. So hybridization is a major threat to biodiversity. It occurs pretty widely in fish, more so than in other vertebrate groups. But genus Lepomus specifically is sort of a hotbed of hybridization. Out of 13 species, we've seen 22 unique hybrids in the wild. And so one of these hybrids occurs between the green sunfish and the pumpkinseed sunfish. Obviously, they created this pumpkinseed green sunfish hybrid. And these 2 fish have a really large area where their ranges overlap, and they hybridize pretty commonly in this range overlap. Specifically in New York, the pumpkinseed sunfish are native to the entire state, while the green sunfish are invasive to a majority of the state. And so this gives this hybrid a unique context in New York, where obviously we have concerns over native fish hybridizing with an invasive fish. Here we can see the shaded region on the left is the native range of the pumpkinseed sunfish, while these points here are invasive records of green sunfish. So there's some prior research out there on this hybrid cross specifically. This Dolly 1987 paper studied the reproduction in an abundant population of these hybrids, and he concluded that these hybrids were sterile and about 99% F1 or first generation hybrids. So this is really interesting, especially when we look at this census data. A couple things I'd like to point out about this. First of all, we see these skewed ratios between either parent species. See the pumpkinseeds are almost 3 times more abundant than the green sunfish. And more interestingly, we see that these hybrid fish are way more abundant than both parent species. And this is not uncommon to see in hybrids that are fertile or sterile, not sterile, viable offspring, where intergression between the 2 parent species is taking place. This is pretty common to see. But obviously this is very odd to see in a population where we know these fish are sterile and first generation. So my project took place on a section of the Old Erie Canal in Fayetteville. This was the original channel of the canal that was dug back in the 1800s. And you can see it curved down through Syracuse and connected to Onondaga Lake. Modern day, west of about Fayetteville, DeWitt area, you can see on that red arrow, has actually been filled in. So it gives us this pretty unique water body where we have a dead-end canal system. Specifically where my study was conducted was at the very end of this canal. So you can see here I have these 2 arrows. This is where I accessed the canal. There's 2 parking areas, and then I collected my fish along the trail that runs up and down this canal. And what I observed here, actually just from fishing, is what led me to this project, is I've been catching all these hybrids for a few years and noticed that these hybrids outnumber both parent species by quite a lot, and also the pumpkinseed sunfish seem to be really low abundance, which surprised me that these hybrids were able to be so abundant, you know, being a sterile fish. And then as I started to look into the literature on this, I found that this Dolly paper had similar findings, and I decided to run my project on this. So I had some objectives to kind of steer me in the right direction. Number 1, I want to know, do hybrid sunfish grow faster than both parent species? We know that hybrid vigor or heterosis is commonly shown in these hybrid fish, and one of these ways this is exhibited is faster growth. So obviously, if these fish were growing faster, this would be key information to understanding these. Number 2, which prey do these hybrids select for? We know that pumpkinseed sunfish and green sunfish generally have pretty different habitat, or different prey selection. So I want to know if these hybrid fish are either selecting for close to one parent or the other, or maybe something intermediate. 3A here I have, could hybrids affect parent species? Since we know these fish are sterile, but they're still outnumbering both parent species, I was thinking maybe there's something going on here. These fish are outcompeting both parent species, something to that. And to kind of look in, and yeah, I came to that conclusion based on, obviously, that observed overabundance of the hybrids from my study slate and the Dolly paper. And kind of zoom in on that a little. I want to know, could these hybrid sunfish affect either parent species disproportionately? We see these skewed parent ratios, so maybe the hybrids are competing with one parent over another. This could be affecting these ratios. So to get a little ID before I go in depth with the methods, Here I have 4 pictures of this pumpkinseed green sunfish hybrid. And if anything, they're probably going to be mistaken for a pumpkinseed, except keys them out for that is they're going to have a more elongated body, much more similar to a green sunfish. They have these really large mouths, again, the trait of the green sunfish, and also these brightly colored fin margins, sometimes almost white, especially on the anal fin, are really good ID characteristics of this fish. So for my methods, I collected 45 fish via angling in June of 2025. My school's collection permit did not extend to this part of the state, so I had to figure something out to collect these fish. At capture, I recorded the maximum gait of the fish. This is the max height that the fish can open its mouth. As you can see my little diagram up there. And then after these fish were euthanized, I collected the stomach contents from each fish, preserved it in ethanol, and then we identified this under my dissecting scope later at the lab. To growth, I took otoliths from the fish and back calculated lengths at age, created these von Bertalanffy models in R, and then also conducted a pairwise likelihood ratio to test each growth model against each other. So getting into my results, here I have max gait. I've got the ratio of max gait to total length on the x-axis or the y-axis. We can think of this generally as the mouth size for each species. And no surprises about the pumpkinseed and the green sunfish. We know pumpkinseeds have relatively small mouths. Green sunfish have relatively large mouths. And these hybrids are intermediate, if that makes sense. And something I'd like to note here is, if anything, these hybrids are much more similar to the green sunfish in mouth size. Well, they are still intermediate. Just something to note that they are closer to the green sunfish. Looking at my diet, here I have the proportion of each prey item grouped by species and proportion related to total contents. And what I'd like to point out here is that both the pumpkinseed and the hybrids selected very highly, or contained very high abundance of snails in their stomach. And as you can see, the green sunfish, not so much. They primarily selected for terrestrial invertebrates. Another way to look at the same data, here I have a prediction of the probability of each fish to contain a specific diet item. So for example, green sunfish, or the pumpkinseed sunfish and the hybrid for snails both have pretty high probability of containing this diet item, while we can see the green sunfish have much lower probability of containing this diet item. Again, with the terrestrial invertebrates, we see pumpkinseed and hybrids looking very similar, about a moderate probability of containing this diet item, while the green sunfish had a very high probability. Every single green sunfish I collected had at least one terrestrial invertebrate in its stomach. And so this was really interesting to see that these fish were selecting for snails specifically. Knowing that these hybrids have much larger mouth size than the pumpkinseed, closer to the green sunfish, I expected them to have a diet more mirroring the green sunfish, but they obviously did not. Also significant is that these fish are selecting for snails, and these adaptations we see adaptations commonly in fish that specialize for snails. Looking at red ear sunfish that are regionally known as shellcrackers actually, and pumpkinseed sunfish, these are two fish that are widely known to specialize in snails. And what these fish have compared to other lipomas is they have much larger pharyngeal jaw size. This is used to crush the snails. And then they tend to have widely spaced short gill rakers. And so if we know these fish have specific morphological adaptations that allow them to feed on these snails, we can maybe infer that these hybrids do too. And if we know they did, then that would strengthen our argument that these fish select for snails. Looking at my von Berg curves, we can see that. So for my von Berg curves, I ran pairwise likelihood ratio test. This tests each curve against each other. I found significant difference in each curve, so they're all different. And then we can also see visually that the hybrid sunfish, the model predicts that these hybrid sunfish have larger length at age size than the other two species. I'd like to point out my output model down below here and the visual difference of the pumpkinseed sunfish curve. I really struggled to collect these fish. I only had 8 samples, and one of these fish was by far and away the oldest fish I collected. You can see it was 8 years old based on those points back there, and also it seemed to have significantly faster growth than other pumpkinseed sunfish I caught. And so my understanding of this K value in L infinity is that the K value isn't necessarily a growth rate, it's how quickly this fish attains the L infinity. And so because we have these points way up at the old age of these pumpkinseeds, they're kind of pulling that growth model to a higher L infinity. Well, they don't change the growth rate per se, so then that K value becomes a lot smaller because it's taking these fish so much longer to reach that L infinity. And again, the low sample size, I don't really trust that output for the pumpkinseed sunfish. So if we look, it might be a little small. This other paper, we have pumpkinseed K value was found to be about 3.68 exactly for their paper. And this was at a similar climate to New York. So even at that range, we can still see that the K values of the hybrids were higher. But I think the most striking thing here was that we can see the model predicts significantly, well, I can't say significantly. Model predicts larger length of age for these hybrid sunfish. So if we revisit my objectives, number one, what prey do these hybrids select for? Snails are the primary diet item of these hybrids, and this really closely mirrored pumpkinseed. So from this, we can infer that the hybrids compete more with pumpkinseeds than they do with the green sunfish. Number two, do hybrids grow faster than both parent species? We know that these hybrids grow faster than both parent species. I'd also like to note that in finding this result, I replicated the results of a very old paper where they were raising these same hybrid crosses in captivity, and they found that the hybrids grew faster than both parent species. So this is really interesting to see that this carries over into wild fish. 3A, we have could hybrids affect parent species? Certainly faster growth gives these hybrids some sort of advantage over both parent species. 3B, could hybrids affect one species over the other? We know this faster growth component, and if we combine this with the fact that these hybrids have similar diet specialization to pumpkin seeds, and also larger mouths than pumpkin seeds, these give these hybrids more advantages over the pumpkin seeds. And from this, we can infer that both of these aspects of the hybrids suggest that they're going to have a greater effect on pumpkin seeds than they do on green sunfish, at least in this system. So alongside my data collection, I collected some samples to do this clearing and staining. My original goal of this was to look for some morphological differences that could aid in identification of these fish. Thinking at young age, some of my identification characteristics probably wouldn't be as visible. So we used two stains for this. Alizarin red stains the bones and heart structures red, obviously. And then Alcine blue stained the cartilage structures blue. And so from this we get these really cool pictures. But what I'd like to point out about this is, well, I didn't collect quantitative data on this. If we remember back to the fact that these snail specialist fish have special morphological adaptations, one of these was these short, widely spaced gill rakers. And I'd like to compare this green sunfish and the hybrid. You can see they have visually different characteristics. And then if we pull an image of this red ear sunfish that we know 100% specializes in snails, these have more similar to the hybrid, certainly. And so we can kind of get an idea that maybe these hybrids do have these morphological characteristics that can allow them to feed on snails. But again, I didn't collect any quantitative data, so maybe this is just something to look into for the future. And so if I could lump up my whole project into one statement, be that hybridization of this pumpkinseed and green sunfish likely has negative impacts on both parent species, but harms pumpkin seeds more. And obviously more work needs to be done to fully understand the effects of this hybridization, but I think this project highlights significant concerns as green sunfish spread through New York and more of the Northeast. And obviously some future studies that I think would be interesting. I think definitely repeat the study on a larger scale in other bodies of water. I know plenty of bodies of water across New York that have these sunfish. It's not clear if they can form these really abundant populations in all of these. Certainly the morphology of these pumpkin seed green sunfish hybrids, again, that gill raker size and the pharyngulate jaw size, both be really good things to have quantitative data to look at. What factors affect hybridization rates? If we could create some sort of list of lakes that might be more susceptible to this hybridization, that could be really useful. Some things that I read in the literature are small bodies of water, skew parent ratios, but there could be more. And also hybridization in other lipomas. We know the northern sunfish can hybridize, I believe, with bluegill and maybe pumpkinseed, not 100% sure. But certainly in a fish that's state threatened, like the northern sunfish, this is even more of a concern. And for my acknowledgements, I'd like to thank Brian Heffron, an assistant with the figures and statistics. Knows a lot more than me about R. I would not have been able to create all these figures without that. Marissa Sup of SUNY Cobbleskill's Histotechnology Lab. She let me use her lab for this clearing and staining procedure, and she was a very big help in getting this procedure to finally work. The SCCA and the Dorman Ham Scholarship for providing funding for this project. Also, SUNY Oswego CSU biologists and the USGS Tunison lab biologists and technicians, I practiced my presentation with them and they provided a lot of really good feedback, helped me get this presentation to what it is right now. So I'd like to say thank you to all those people. And if you want to contact me, I'm currently working as a technician for SUNY Oswego. I'm stationed at the USGS Tunison Fish Lab in Cortland, New York, and you can reach me at my email here. But with that, I'll turn it over to questions if anyone has. Find me afterward.
Find Will afterwards, no time for questions.